Hey Team Bio, welcome to your screencast on anaerobic cellular respiration. Okay, quick note, we've talked about this overall graphic, and this is a flow chart through all of cellular respiration. There are two pathways. One pathway is the anaerobic pathway, and this is done in the presence of, or I should say, in the absence of oxygen. And the other is the aerobic pathway, which is done in the presence of oxygen. They both start with glycolysis. So, anaerobic cellular respiration is equal to glycolysis. Plus fermentation. And aerobic cellular respiration is equal to glycolysis plus prep steps plus Krebs cycle plus, what am I calling it here? Electron transport phosphorylation. Okay, and today we've talked about glycolysis. So this is where both pathways start. It occurs in the cytoplasm. And today we're going to be talking about fermentation. This additional pathway we tack on to complete the anaerobic pathway. And so that's it. Today, screencast is about fermentation. Okay, so glycolysis, as you know, begins with glucose. And the point of cellular respiration is to slowly combust glucose. We're going to break it down into CO2 molecules. And in doing so, we are going to release a bunch of energy. Some of that energy is going to be trapped directly in the form of ATP. So last screencast, you learned that we did some substrate level phosphorylation to make 4 ATP, but remember, we spent 2 ATP at the beginning. So on net, when you subtract the 2 that we made from the 4, we get 2 ATP made. We also performed a redox reaction. So we had this 3-carbon molecule, G3P, that was charged up with some energy that we deposited on it from ATP. And we extracted some high energy electrons. So these were HEEs, high energy electrons. And we stored them on a carrier molecule called NADH. So when electrons are transferred onto this, we say it is reduced. And G3P was oxidized because it lost electrons, and NADH gained electrons. Okay, so we also made, in this reaction, 2 NADH, which are carrying our high energy electrons, remember, which are kind of dangerous if we let them loose in the cell. And then the last thing that comes out at the bottom is 2-pyruvate. And these are the 3-carbon compounds that were once glucose that now have been slightly changed in form, slightly reduced in energy level, and we'll proceed on to the next steps. So we're going to continue to oxidize pyruvate and extract energy from it. But actually, that only happens in the aerobic pathway when we have the presence of oxygen. If we don't have oxygen, we have to proceed down the anaerobic pathway into fermentation. And fermentation actually creates no useful cellular energy. And you might be wondering, why do we do fermentation if no useful cellular energy is produced? And the reason is good. We recycle NADH back to NAD plus. And by recycle, I actually mean we oxidize it. And this allows glycolysis to continue. And let me explain what I mean by that. Okay, so glycolysis, we put in some glucose, we put in 2 ATP, we get out 4 ATP and 2 NADH, and some pyruvate. So this is useful to have 2 ATP. This is useful if we have oxygen, and this is also useful if we have oxygen, and also if we don't, we're going to use it to recycle the NADH. Glycolysis can't keep working unless there is a free supply of NAD plus here. This step, moving from G3P to pyruvate, requires the presence of NAD plus. And if all the NAD plus in the cell is booked up, holding electrons and it's NADH, glycolysis can't proceed. And that means we are not making ATP. Even though it's just a small amount of ATP, it's better than nothing. So the point of fermentation is to recycle NADH, free it back up into NAD plus by oxidizing it and allowing glycolysis to continue. So let's talk about how that happens. Okay, the inputs to fermentation, regardless if you are an alcoholic fermenter or a lactic acid fermenter, are the same. They both start with pyruvate. Oops. And remember, different types of, okay, well, I guess we'll do a straight line. Different types of organisms either do alcoholic fermentation or they do lactic acid fermentation. So you are a lactic acid fermenter. This means when you go run around really hard and get out of breath, you're producing lactic acid, not alcohol, ethanol. Yeast, however, are alcoholic fermenters. And so when you confine them in a space with limited oxygen, they will produce a bunch of ethanol. And we'll talk about how that happens. Okay, let's start with alcoholic fermentation. So alcoholic fermentation is a little bit more complicated than I'm drawing here, but we're just going to focus on the carbons, as I said before. Okay, so it starts with 2 molecules of H2O. Each one of these, maybe I should just draw them as 2 independent molecules, is going to come in here and break up this 3-carbon compound, performing a hydrolysis reaction, and spit out these carbons here. So these carbons are actually going to peel off and we're going to be left with a 2-carbon compound called acetaldehyde. Not sure if I spelled that right. Let me double check. I didn't. GACETALDEHYDE. Okay, so this is a 2-carbon compound. It has a strong fruity odor, actually, and it is the precursor to ethanol. What happens next is that the NADH, I wonder if we should draw, that was produced in glycolysis. Let's turn this to red. Let's make it like this. Is going to swoop down in here and drop off some high energy electrons and some H+. And in the process, so this is NADH coming in, it itself is going to be oxidized and recycled back to NAD+. So acetaldehyde in this case is reduced. Because it picked up electrons. And then ultimately, out of that reaction, we get ethanol. Okay, so we performed a couple different things. Number one, we've made some CO2 in this process. So if you ever let something go bad in your fridge, in a jar, you might notice that it's a little bit bubbly. That is CO2 likely caused by yeast, a fungus, performing fermentation in your fridge. It also probably smells maybe a little fruity and a little alcoholic. And that is because ethanol is another product. The whole point of this is to free up NAD+, so the yeast can continue to run glycolysis and get out 2 ATP. Not very efficient, but hey, it works. Okay, the next example is what humans do, and also the bacteria that are in your yogurt. Okay, they start with pyruvate, just like the yeast in alcoholic fermentation. So the pyruvate enters, and it's also a little bit more complicated than this, but we're not going to really get into the nitty-gritty here. And once again, the whole point of this reaction is to take the 2 NADH that were produced in glycolysis and use them to drop off electrons, these high energy electrons that are kind of dangerous. So this is NADH, and safely get rid of them to oxidize NADH back to NAD+. And in the process of dropping off those electrons and also some hydrogen, out at the other end pops lactic acid. Now, you might notice that yogurt, for example, is like a little bit tart, a little bit acidic. That is because this is a weak acid, this lactic acid that is produced at the end of lactic acid fermentation. So once again, this compound is reduced, and our NAD+ is oxidized. And the whole point of both of these reactions is to free up this NAD+ so it can return to glycolysis and do its thing, accepting electrons from G3P up here. Okay, that is all. That is fermentation. We're going to be setting up an experiment in class on Monday of Tuesday when you get back from break. And we're going to be measuring the rates of fermentation in yeast. So think about what type of respiration, anaerobic cell respiration are yeast doing, and what products should we expect to see and maybe smell. Okay, that's all.